So we're talking about DNA replication, and first we need to talk about what is the purpose of replicating the DNA. So why do cells need to do this? Cells are going to carry out DNA replication to prepare for cell division. Oops, not cell divisions. Cell division. Okay. So in order for a cell to make more cells, it has to first replicate its DNA, and then it's going to go through cell division. We're actually going to talk about cell division next week. We're going to talk about mitosis and how that works. And when we go through cell division, each new cell needs its own DNA. So we replicate our DNA, and then we have 2 copies of the DNA, and then we give 1 to each of our new cells when the cell splits up into 2. So they replicate their DNA first, and then they divide. So we're talking about how they replicate that DNA. Next week we'll talk about how they divide and how that process happens. Now we call DNA replication semi-conservative, and the reason for that is that the new DNA is composed or made up or created of 1 strand from the original parent DNA, and then the other strand that's going to be newly synthesized or created. So we say that DNA replication is semi-conservative because when we create our new DNA, half is from the original strand and then half is brand new. So 1 piece is from the parent DNA, and then 1 piece is the newly synthesized. And if we go look over here on our picture, we can kind of color code this. So we'll say that our parent DNA is purple. So both of my strands should be purple because this is my parent DNA over here. Something like that. Okay. Then when we replicate it, we're only going to have 1 purple strand in each of these. So I'm just going to color 1 of these as purple. So there's our 1 parent strand for 1 of our new DNAs, and then here would be our other parent strand for our other DNA. And then our other strand would be our newly synthesized one. So we could either do that in a different color, you could leave it blank. So this is why we call this process semi-conservative, because only part of the original DNA is conserved in the new DNA. We split it up. Now let's talk a little bit about some of our important enzymes. There's a lot of different enzymes and proteins that go into this whole thing. So these are kind of like the 6 big ones that are used all the time, but there's even other smaller ones too that go in and do different things. So our first one is our helicase. This one's kind of the one that will come in first. And it acts kind of like a zipper. So this enzyme will unzip the DNA like a zipper on a coat. So it helps break apart the DNA into 2 separate strands. So it creates 2 single strands of DNA. So it takes the 1 double strand of DNA and it unzips it into 2 single strands. So helicase kind of acts like a little zipper, unzipping our DNA. If our DNA is still zipped together, nothing else can happen. So this one has to usually act first. It'll go through, it'll unzip the DNA, and then all of the other enzymes can then kind of fit in there and do their thing. Shh.
Riley.
I am.
Respect.
Mm-hmm. Our next enzyme is called topoisomerase. So this is kind of a longer one. But this is kind of the muscle of the operation. It's the muscle. So topoisomerase keeps this DNA from coiling up super hard. So it relaxes the supercoiled nature of DNA. If we didn't have topoisomerase, our DNA would kind of curl around itself and then tie itself into a big ball or a big knot, which would not be good. So topoisomerase helps relax the DNA so that it doesn't coil up, and that way we can still unzip it. It says add some muscly arms, so do your best. Ooh. Bam. Not very good. That's okay. So our topoisomerase is the muscle of the operation. It helps relax the DNA. So in that video, it kind of showed it. It's usually right by where we're unzipping it. So the topoisomerase and the helicase are usually pretty close to one another. The topoisomerase helps relax the DNA right before our helicase zipper gets there to unzip it. Now our primase acts like a flag at the start of a race. So this enzyme helps prime the DNA, and it creates a starting point for DNA polymerase. So it kind of tells DNA polymerase where to go and says, hey, this is where you're going to start making more DNA. So it adds kind of like a little flag of this is where you should be. Kind of like a primer. That's why it's called primase. We give him like a little flag. So our primase primes the DNA. It's kind of in its name. That one's pretty easy to remember. And it tells the DNA polymerase where to start. Now it tells the DNA polymerase where to start. Our DNA polymerase is one of the most important ones. This one is actually what's creating the new DNA. So this builder enzyme works hard. It's synthesizing or creating new strands of DNA by adding our DNA nucleotides. So our DNA polymerase is the one that brings in those extra nucleotides and is actually creating the new DNA strand. So far everything else has just been kind of preparing for that. DNA polymerase is the one that actually comes in and starts to create that new one. So he's the one building DNA. Says add a little builder hat. Give him like a little hammer. I don't know. So the DNA polymerase is the one that's building that new DNA. It's gorgeous. I like it. The exonuclease and the ligase, they talked a little bit about the ligase in the video, but the exonuclease is one that they didn't talk about. So the exonuclease acts kind of like a pair of scissors. So it's going to cut out RNA primers and DNA that needs fixing. So our DNA polymerase has some proofreading capabilities. So it's usually that our DNA doesn't get messed up when it gets copied. Now, just in case it were to, our exonuclease will go through. It'll make sure that it cuts out all of the RNA primers that we don't need anymore. And it'll also fix any DNA that needs a little bit of fixing. So if DNA polymerase were to accidentally mess up, then our exonuclease helps fix it. So we'll give this one like a little pair of scissors.
judging my scissors.
Yeah, go show Bailey your scissors.
You show me your scissors later.
I don't want to.
So our exonuclease cuts out anything that we don't need before we package it up. And then the last thing we have is our ligase. So this new DNA strands have breaks between fragments on the lagging strand. So there's little gaps that we don't want there. And so the ligase helps join them together like a glue stick. And so it helps join the DNA together and stick it all together. Otherwise, our DNA would be in a bunch of chunks, which would not be good. So it acts like a little glue stick. And it glues all of those fragments together. And on the other side, we'll be able to see those breaks on the lagging strand. Okay. So once you're good with this side, you can flip it over. We'll do it together. Okay, are we all good with that side? Make sure you at least have all of the blanks down. Okay. On the other side, this is what it looks like if we were to zoom in really, really far to what else is happening. So we're going to go through, we're going to label all of our different enzymes that are working here and all of the different parts. And we'll also go through the different steps. So we'll do the steps first and then we'll label everything. Our first step is where we unzip. So that's when we have our enzyme helicase, which we talked about on the other side. Helicase is going to come in and unzip our double-stranded DNA to create our two single strands. How it does that is it goes in and breaks the hydrogen bonds that are holding the DNA together. So when we talked about our nucleotides, we talked about how hydrogen bonds hold together our cytosine and our guanine and our adenine and our cytosine. No, I already said that one. Thymine, that's the other one. So hydrogen bonds are holding those together. Helicase will come in and it'll break up those hydrogen bonds. It'll unzip everything and it creates complementary, or it breaks it up between complementary base pairs, separating it into single strands. So we go from one double strand to two single strands. And then our helicase enzyme is this one right here. So you can see it's kind of going in where the DNA is still joined and it's breaking it up as it goes through, kind of unzipping it. So this is our helicase. Now, while we are unzipping our DNA, it can get a little unstable. So we do some things to stabilize it. So SSB binding proteins are attached to the unzipped DNA. We can see them right here. So these are our single-stranded binding proteins. So they attach to the unzipped protein here, and it stops it from joining back together. Those DNA pairs like to be together, and they will choose to be together unless we put something there to keep them apart. So our SSB proteins will help separate those. And then we also have an enzyme called topoisomerase. It's a long one. But our topoisomerase relieves tension in the DNA. It relaxes it so that it doesn't supercoil. So it's relaxing it from its supercoiled structure. So the SSB proteins help keep the nucleotides together or apart. And our topoisomerase helps relax the DNA so that it doesn't coil up really hard. So on our picture, this kind of donut shape here is our topoisomerase. So it kind of wraps around the DNA, helps relax it.
Oh my gosh, baby.
You already wrote it once. I believe in you. It's a long one, I know.
You did it again.
And then here, these little kind of oval shapes, these are our single-stranded binding proteins. Binding proteins, or SSB proteins. So they shorten it. So they bind to a single strand, single-stranded binding proteins, and they help keep the DNA separate, so it doesn't try to join back together. Otherwise, our helicase wouldn't be doing a very good job. They would be separating the DNA, and then it would be instantly joining back together. Now, once we have kind of separated our DNA and we've stabilized it so that it won't join back together, we've relaxed it so that it's not going to supercoil, we have to prime it. So we use an enzyme called primase. Our primase is going to attach to our RNA primers to the DNA. So it provides a starting point for DNA polymerase. So this is like our little race flag. It kind of tells where our RNA primer needs to go to create our new DNA. And so in our picture, it's right here. So we can see it attaches to the start of where we want to create our new strand of DNA. So right where it's unzipped is usually where this guy's hanging out, real close. So this is our primase. So this one creates a starting point to create more DNA. Now we're going to go up to step 4 at the top middle for how we actually build our DNA. So we have unzipped it, we've relaxed it, we've kept it from joining back together, and then we've primed it and we said this is where you're going to start. Now we actually have to build it. So our DNA polymerase, which is the builder enzyme, is going to join up with the DNA, and then it helps build the new DNA strand. So our DNA polymerase associates with the DNA or joins with the DNA and helps build a new DNA strand by adding our DNA nucleotides. Now there is a catch with DNA polymerase, and that is that it can only build a new strand in the 5' to 3' direction. Now our DNA has directions. We can have a 3' end here to 5', 5' to 3'. They always join opposite. And so DNA polymerase only works one way. Because it only works one way, we end up creating a leading strand, which is usually on the top here. This is our leading strand. And then we create a lagging strand, which is usually on the bottom. Our lagging strand has these gaps. So that's how you can usually tell the difference. The leading strand doesn't have any gaps here. Lagging strand has these weird empty spots. And then in our picture, our DNA polymerase is this guy, so it's the one that's kind of here and adding new nucleotides as it goes along. Or here it's adding new nucleotides as it goes along. So this is our DNA polymerase. And then we'll label our two strands. The one at the top I said is the leading strand. That's the one that doesn't have any gaps. That one, our DNA polymerase can just keep going along. It doesn't need to stop. And then the bottom here, this is our lagging strand. Our lagging strand, the DNA polymerase kind of has to work backwards, so we get these weird gaps. Now, after we are kind of all done adding all of our nucleotides, we have to remove all of the primers that we used. So the primers have to be removed by exonuclease. So it comes in.
Why is it over here?
The primers are removed by exonuclease. We don't need them anymore. After we've created our DNA, we're kind of all done with them. These are made of RNA. And if we're creating DNA, we only want DNA left. So once we're all done with our RNA primers, DNA polymerase is going to come in and replace them with DNA nucleotides. So we don't want to keep any RNA with our DNA strands. That might make things messy when we have to go make genes and cells later. So DNA polymerase is going to come in and switch them out.
Riley, come on.
Riley, stop touching the lamp. Leave it alone. Okay, so RNA primers would be like this guy here. This is an RNA primer. So the RNA primers are necessary in the beginning, but then we take them out and get rid of them later. After we have replaced all of our different RNAs, we've gotten rid of them, replaced them with
Does that
DNA.
say RNA primer?
Yeah. After we've gotten rid of all of our RNA primers and we've replaced them all, we have to glue our strands together. So you can see we have these weird gaps that'll happen. And we don't want that with our DNA, so we glue it all together. So we have ligase that will come in and it joins or glues the DNA fragments together. So our ligase acts kind of like a glue stick. It's going to go in and it's going to join all of those small fragments together. We call those fragments Okazaki fragments.
What?
You're not making us write that, are you? What?
Nah.
Motherfucker, Bailey.
Oh, it's not.
It's only 7 letters. It's really not too bad.
Oh, yeah, never mind.
I suppose it's not looking.
Yeah, so these are called Okazaki fragments. That's their actual name. If I were to guess, I think it's named after the scientist who discovered them, would be my best guess. Or it has some other meaning. I'm not quite positive. But they are called Okazaki fragments. That's their official name. It's kind of a weird one.
No.
And then our enzyme ligase is the one that's gluing them, and so we can see our ligase over here. So the ligase is kind of gluing these things together to make them all one nice strand. So you can see up at the top here, We don't really have any Okazaki fragments. We don't really need to use the ligase at all because we don't have any gaps. Down here you can see where we have these little fragments and we have to stick them all together. So ligase will come in and stick them together.
You're welcome.
And then our very last step is our proofreading step, just to make sure that everything looks good and there's no mistakes. So another type of DNA polymerase. We'll scan the DNA, make sure that there's no errors. If there are errors, it'll then fix them. So it scans the DNA for errors and then fixes any that are found. So it's important that our DNA doesn't have any errors, otherwise that could lead to mutations, which could then lead to things like cancer and tumors, which would not be good. We don't want that.
That's not good.
That would not be good. So DNA polymerase will come in, make sure that there's no errors, and if there is, then it's going to fix them before we have problems, which is good. We don't want problems. And then I think I missed 2 blanks when we were talking about them. This one, these are our DNA nucleotides.
Wait, what?
DNA nucleotides, these little like
Oh,
T
shit,
things.
I put the wrong thing in.
So the DNA nucleotides are what makes up our DNA. Those are all of those individual bases. And then this one over here is our exonuclease, which we talked about. But our exonuclease goes in and removes the primers. So you can see here, it's kind of taking out the primer and putting in a DNA nucleotide.
Are we done?
We are done. If you would like to color them, fill them in, label things,
What do we do with these?
they are your notes. Hold on to them for a future test.